morning friends. So, today we have come to the concluding session of this the entire fluorescence spectroscopy lecture series. So, actually we would like to sum up what we have tried to learn in these series of lectures, starting from very basic principles to various applications, etc., etc. And also in this particular piece of, in the final session, I would like to acknowledge various books. Because books will be are very useful in this regard, and also various website informations. In many cases we try to borrow many informations, maybe in some cases we made photographs, or maybe we made some diagrams based on the ideas obtained from those websites. So, in appropriate places we try to acknowledge those website informations. Nevertheless, importantly, in some cases Wikipedia informations are very useful. Maybe various journals are also very useful, like Journal of Physical Chemistry, Journal of Photochemistry. Then there are plenty of journals available. Then many biological journals. These are also very useful, in making this study material and lecture notes. Now, throughout this series of lectures, we started with introduction, where we tried to introduce to this subject, like what is exactly the science behind there, and it is basically an interaction of interaction of matter with radiation. In this case, matters maybe atoms, maybe molecules, maybe materials, and so on, and maybe biological samples, etc., etc. And we try to gather information based on the information that is based on fluorescence information, that is the molecule emits, and we collect this emission information and then analyzed based on whether it is a time domain information or it is a frequency domain information, and also analyzing those informations. We try to make a link between what exactly the molecular phenomena or the atomic phenomena or the physical phenomena that are involved involved in the process. Okay, so involved in this light emission processes. Okay, now we talked about what is spectroscopy. Okay, spectroscopy means it is nothing but the ordered arrangement of something, and here as a function of wavelength we generally make this ordered arrangement as a function of wavelength. Maybe it's a fluorescence intensity, in some cases maybe extent of absorption, or maybe extent of the light that is transmitted through the sample. Okay, so and we also talked about various photophysical processes. Photophysical processes, like the process which are physical in nature. There is no chemical transformation actually occurring during this photophysical processes. That means it is simply absorption of light, then system is getting excited to some upper electronic level. Okay, and then from this electronic level it is undergoing various physical phenomena, maybe like non-radiative deactivation, maybe radiative deactivation, maybe intersystem crossing, maybe internal conversion, maybe fluorescence, maybe phosphorescence, maybe heat. So this consultation with this Jablonski diagram will help in understanding all these photophysical processes. And maybe in any of the electronic excited states, the molecule may undergo chemical reaction, as a result of which new entities are formed, maybe some photochemical reaction that will lead to formation of something else. Okay. So various photophysical processes we talked about. Then we also talked about what is luminescence. It is a general term. Then from that we move to fluorescence. What is fluorescence? It is exactly basically it is a emissive process in which this this electronic this spin part. Okay. So spin state is conserved in this process, that it is a singlet to singlet transition. It's a radiative singlet to singlet transition. and also we talked about a little about this phosphorescence. This is another kind of luminescence which is slower in terms of time dependence. It takes time because it is a spin forbidden process between singlet and triplet levels. Since singlet and triplet levels are no longer remaining pure state under spin orbit interaction scheme, this pure singlet is converted to a mixture of singlet. It's a combination of singlet and a triplet. Therefore, a pure state description is not a good approximation. So that description fails, and people try to explain in terms of this mixed state where some fraction of this triplet character is coming into this originally singlet state, and as a result of which this pure this singlet to triplet transition, okay, in contrast to mixed triplet singlet. So this process is coming, and therefore mixed triplet to another mixed singlet triplet to another mixed singlet triplet state, this transition is taking place, radiative transition, and that leads to this phosphorescence. And this phosphorescence, it is a very much a redshifted emission, and it's a slow process compared to fluorescence. And we also talked about that, and various methods of detection. We also have talked about that. Now, whenever it is the question of luminescence, that is fluorescence, then the question of this quantum yield and lifetime comes into action. So we also talked about what is lifetime and what is quantum yield. That's a very important quantity. Okay, so quantum yield gives you many information with respect to the various processes that are taking place in the process, and taking place in this various photophysical or maybe photochemical processes. Okay, so we talked about lifetime, then we talked about fluorescence, quantum yield, then we talked about fluorescence anisotropy. Fluorescence anisotropy is a very important thing, that is because of the anisotropic distribution of this solute dipoles as a result of excitation, and therefore the moment the excitation is going out, I mean when the excitation pulse enters into the sample, and maybe some of these photons are absorbed and remaining is exiting your sample, then the molecule starts to in the excited state starts to rotate in various fashion, and as a result of which what is happening, that maybe some of the dipoles are oriented in this way, some of them are oriented in this way, that is polarization wise, I mean with respect to this incident polarization, and therefore some of them are intermediate. Therefore this as a function of time, the average number of dipoles that are oriented in a particular direction, it is changing, and therefore this emission that is collected either in this parallel or maybe perpendicular configuration in the analyzer side, you will be finding that it is also behaving differently, and that information gives you regarding the environment where this solute is located, whether it is facing more rigid environment or maybe less rigid environment, whether the cavity where this fluorophore is located, that is facing some other kind of interaction or not. If there is interaction like hydrogen bonding interaction, maybe this depolarization or maybe movement of these dipoles will be restricted, and result of which it will be this motions will be slowed down. So that that dealt with this anisotropy. We talked about instrumentation in a great detail. Okay, so what is there in the instrument, what are the parts, etc., etc., and what are the roles of this individual parts, that we talked about in this instrumentation. Like, you know, we talked about little about this absorbance spectrometer. Then we talked in detail on this fluorimeter. Okay, and what are the parts of fluorimeter. Okay, and how this you know, emission is collected, like what is the reason for collection of emission at 90-degree geometry, that we also talked about. I mean, just to reduce this excitation wavelength. Okay, I mean the intensity due to excitation light. So these are the things. And then what are the type of detectors that you would be looking for while, you know, thinking about some instrument. So you can fabricate your own instrument, but you have to be very careful about what you are trying to look at, whether it is a high resolution you know, instrument, or it is a low resolution instrument. So, and that is determined by the dispersion optics. So if you want to use a good dispersion optics, then your system will be you know, will have high resolution. Otherwise resolution will be compromised because of the compromisation of this dispersion optics. Okay, so we talked about this fluorescence polarization and their implications and their possible applications. Then we talked about a very important issue of this fluorescence quenching. Now, what is fluorescence quenching? We talked about various models of fluorescence quenching, like it is a dynamic model, or whether it is a static model. Dynamic model means it is within the lifetime of the excited fluorophore, the quencher is hitting the excited fluorophore, and therefore the energy is lost, and therefore its fluorescence quantum yield is reduced. On the other hand, in case of your the other mode, that is, you know, ground state, when there is a ground state interaction, therefore in the ground state already there is interaction between your unexcited fluorophore plus your quencher, and therefore, you know, free fluorophore which can be excited, which will give rise to emission, will be reduced, because some of them are already involved in forming a dark complex, okay, which is not able to fluoresce. Okay, so that's why this other quenching mechanism is there, and this quenching mechanism, you know, these two quenching mechanisms are very useful in diagnosing various interactions that are taking place at molecular level, like maybe interaction between your fluorophore with some quencher, or maybe interaction of a you know, species means maybe it's a protein, maybe some material contains fluorophore. So whether there is interaction between this quencher with maybe this protein, so whether there is an interaction or not, and what is the nature of interaction, whether it is electrostatic, non-electrostatic, or something else, whether it is a hydrogen bond type of interaction, or whether it is a, you know, it is a dynamic quenching mechanism. So that is you know, very nicely, I mean, it is discussed in detail. And this we also talked about this Stern-Volmer expression and various an implication of this Stern-Volmer expression. Okay, then we talked about energy transfer and electron transfer. So energy transfer, criteria of energy transfer, and what is the implication of the study of energy transfer. And we talked about the spectroscopic ruler, that is, using the spectroscopic principles, you can find out the distance between your energy donor and the energy acceptor, and what are the factors that are responsible for altering this the flow of energy from the donor to the acceptor, that we also talked about. We talked about mechanism like Foster type of energy transfer mechanism, Dexter type of energy transfer mechanism, and their possible implications, and you know, this what are the factors, like whether how geometry factor is important in ensuring this energy transfer from donor to acceptor, whether means what is the geometrical requirement, what is the angle between, what should be the angle or how this angular separation between the donor dipole and the acceptor dipole is is determining the extent of energy migration from donor to acceptor. Is there any solvent effect or not, that we have talked about. Is there any effect of temperature, etc., we talked about. Then we talked about electron transfer. That is a very important phenomena, that electron transfer, and also this electron transport is a very important phenomena, especially in biological system, like in mitochondria. There is a chain of electron transport. Okay, ultimately that is giving rise to energy. I mean, it gives out energy, and using that energy we are able to do our work. So electron transfer is a very important phenomena, and whether this electron transfer, I mean, what are the factors that are responsible for controlling the electron transfer between the electron donor to electron acceptor. What is the role of this this reduction potential of these two these two species, like one is electron donor and another is the electron acceptor, and what are the factors that can affect this rate of electron transfer. Okay, and we talked about this Marcus theory of electron transfer and various factors that are responsible in controlling this electron transfer, and also we talked about the importance of electron transfer in various chemical processes. And this electron transfer actually in fluorescence spectroscopy, the electron transfer process which we are talking about is either the donor or the acceptor, one of them is in the electronically excited state, and there is a transfer of electron. And actually energy transfer may occur or is known to occur in ground state also, like in regular oxidation reduction process in the ground state. Then we talked about the effects of environment on fluorescence emission and fluorescence lifetime. So that we have talked about, means how this environment is affecting the emission quantum yield and also this lifetime, and what is lifetime and how this lifetime is affected as a result of environment. That is the environmental effect on this fluorescence emission quantum yield and lifetime. Environment means where, I mean, the environment, we are talking about the environment where this your fluorophore is located. Okay, whether it is in aqueous medium, whether it is in a non-aqueous medium, whether it is less viscous media or it is a more viscous, whether there is a possibility of hydrogen bond, or whether temperature has got any influence on these parameters, that is lifetime or maybe emission quantum yield. Okay, so these we talked about in detail. Okay, next we talked about this exciplex and excimar. Okay, exciplex means excited state complex, excimar means excited state dimer. Okay, so what is exciplex, how this exciplex is formed, and their energetics, and then exciplex means it is excited state complex where you are generating two unpaired electrons. Okay, and this exciplex can be in singlet state, it can be in triplet state, and so how the external factors like a magnetic field can affect this exciplex luminescence, because exciplex is a broad has got a broad emission spectra. So how this emission quantum yield gets affected due to environmental factors as also due to this external perturbation like magnetic field, external magnetic field. Okay, this we have talked about in detail, and with the help of this vector atom diagram, okay, we tried to explain what is happening in case of a singlet exciplex and in case of a triplet exciplex. We also talked about this excimar and this how this excimar and also its energetics and also its potential energy surface, and excimar the concept of excimar in making excimar laser. Okay, so we also talked about this excimar laser. Okay, Now we talk, as I told you this separately, we talked about this magnetic field effect on exciplex luminescence. Okay. So magnetic field can affect the spin chemistry. So it is basically exciplex is nothing but spin chemistry. So how spin chemistry is getting affected in presence of external magnetic field, we talked about in great I mean it's a we talked about a lot on that. And also one thing that I should mention over here, that this not only in case of exciplex, but also in other type of processes like hydrogen abstraction process, or maybe this recombination of two radicals, they are not only on luminescence but also on absorption process. The magnetic external magnetic field has been found to affect their recombination kinetics. Recombination means when the back electron transfer is taking place, and then the donor is accepting back the electron which was already given to an acceptor. Okay. So donor accepts back, that is called as back electron transfer. Okay. So that part is although out of scope, but it can have good correlation with this magnetic field effect on exciplex luminescence. But the thing is that the mechanisms are a little bit, the mechanisms means the basic thing is basic spin chemistry is same, but the effects etc., these are explained in with the help of different models. Now then we talked about metals that are influencing fluorescence. Okay. So metals have got tremendous importance in controlling fluorescence. So we talked about that, and then after that we talked about this chemiluminescence, then a little idea about bioluminescence. So chemiluminescence means chemical reaction, and then this chemical reaction leads to luminescence. So we talked about, so this luminol, then luciferin, luciferase, etc., we talked about. So we gave a good introduction about this chemiluminescence and its importance and also its importance. Then we talked about a very important aspect of this fluorescence studies is the solvent relaxation, because we all during our talk, during the series of lectures in earlier lectures, we talked about that fluorescence is very much dependent on the environment. So solvent relaxation is basically when you are creating an excited dipole, then around this excited dipole, before creation the solvent molecules were organized in a different fashion. Then after it is created, then this reaction field is changing, and as a result of which this solvent molecules will move, will rotate themselves in such a way that it gives you energetically a better situation for your this fluorophore. And taking the advantage of this fluorescence measurement and also this time dependent fluorescent measurement, that is especially this with the advent of ultra fast methods, and also this single photon counting devices. Okay. In single photon counting devices, you count photons that are emitted from your sample. Sample means fluorescent sample, and counting that and then putting in a histogram, you can find out this decay time. So the point is, if you change this environment, that is solvent environment, then this fluorescence parameter like decay parameters etc., these are going to be affected. Had there been any effect of this environment on this fluorophore itself, that is, if the fluorophore is facing a different environment as a function of time, and that can be monitored, and if it is facing, and if the fluorescence property of your fluorophore is changing as a function of this environment, then in principle you can probe that, not directly but indirectly, by probing the time dependent Stokes shift of this of your fluorophore, and just by monitoring that, as a function of time, you should be able to look into solvent relaxation process. So it is a very important study, and you will be finding that there are various researchers, not only abroad but also in India. They are involved in this studies of solvent relaxation, which is broadly called solvation dynamics. So that's a very interesting field, and it is a very matured field. Okay, now next we talked about ultra fast studies. Ultra fast means we talked about the various domains like fast, then ultra fast, then slow, then steady state, so on and so forth. And in ultra fast study, what is the basic motto of ultra fast study? Actually, dynamics of solvation is also a part of this ultra fast, also in the ultra fast domain. So ultra fast study means any process which are occurring in ultra fast time scale, maybe in picosecond, maybe in femtosecond, or maybe in sub femtosecond. So these ultra fast studies are very important, especially this photon mediated processes, and some of them are known to be very important. Some of them are and a number of them are known to be very important, especially in biological context. Okay, so actually in our vision system, this first process, that is this cis-trans process, I mean this geometrical rearrangement is a very crucial step in our vision cycle. So we talked about that also. Then we moved to protein fluorescence. Okay, we moved to protein fluorescence, and in this heading, we are still not in imaging. We have not still talked about imaging until this protein fluorescence. Okay, so here significance of protein fluorescence, we talked about that, and then what are the factors that are responsible for this intrinsic fluorescence of protein. Whether intrinsic fluorescence of protein is the last answer, or you can add one fluorophore from outside which will be able to see something. So that we talked about, and then how we are going to employ this protein fluorescence in our various studies, and we gave a number of examples where this protein fluorescence has been utilized. Protein fluorescence, and also fluorescence involving protein containing systems. So where we have shown that using this protein fluorometric studies, we can look into various processes like interaction between biomolecules, interaction between a biomolecule and a drug, or maybe some small molecule, and what are the factors that are responsible for the stability of such complex, that is after this interaction, I mean due to interaction some complexation is happening. So the factors that are responsible responsible to stabilize this protein then ligand complex, and what are their implications, I mean how this can be important in our scientific scientific domain, how these are important. So we talked about all these issues over there in detail. Then we also talked about this when it is the question of fluorescence measurement, then naturally this time resolved studies are very important related to protein fluorescence, because protein is a very complex system, and you can find a globular protein, you can find a fibrous protein, maybe a mixture of these different structural features. So because of this molecular interaction, or maybe change of pH or some other factors, this structural features are getting affected. So when structural features are getting affected, then naturally the site which is responsible for fluorescence, like this fluorophore, intrinsic fluorophore, that is also getting affected. So so that means the environment is getting affected as a result of maybe protein ligand interaction, or maybe structural modification due to change of pH, maybe due to change of temperature. So that you can actually follow with the help of this not only steady state but also time resolved studies, where a number of important aspects can be can be understood, can be looked into. Okay, so that we talked about under the heading of time resolved studies related to protein fluorescence. Okay, then we have come across this a new issue, that is photoluminescence, and also importance of fluorimetric studies or maybe luminescence studies, okay, relating to materials, because nowadays you will be, if you go through different journals, you will be finding that plenty of scientific papers are coming every day, are published every day on materials and material science. And okay, so and whenever there is a material, then maybe some materials are fluorescent, some materials are not fluorescent, or maybe you can you use this fluorescence technique to use this material for various applicative things, like maybe sensing of molecules, etc., etc., sensing of small molecules, or maybe some other in some other diagnostic purposes. So in this particular piece of lectures, we talked about what is fluorescence, what is the connection between fluorescence and photoluminescence, and how this fluorescence and photoluminescence is important as far as materials materials are concerned. And in this material we talked about this, especially nanomaterials. So it is the era of nanomaterials and quantum dots. So we talked about materials, how this material fluorescence means fluorimetric studies involving materials can be very useful in many applicative things. And also we talked about quantum dots. Okay, quantum dots are new materials, okay, which are having fascinating properties, and employing that we would be able to, people have shown that, scientists have shown that those materials can be used for various optical, then various electronics and various chemical problems. Then we talked about this fluorescence microscopy. That's in last 3 lectures we dealt with this fluorescence microscopy. What is fluorescence microscopy? What is the relation between microscopy and fluorescence microscopy? Why we will be interested in this particular thing, like in this fluorescence microscopy? So that we have talked about, and then various type of studies we also talked about in fluorescence microscopy. So, and we gave many representative examples, and also we gave detailed account of these various parts, detailed account in the sense that we tried to give you an overview of a fluorescence microscope, its modification. Then we talked about this confocal microscopy, because confocal microscopy is the latest technology in case of fluorescence microscopy, and it has got tremendous application nowadays, especially in medical field and also in materials field. These have got tremendous applications and use. And nowadays you will be finding, with the advent of this newer technology, newer technique, now this microscopy thing has become very interesting and useful. And maybe you will be finding that a number of research groups, not only abroad but also in India, they are involved in this study using fluorescence microscopy. We talked about various techniques, and like for fluorescence microscopy and their various applications, and we talked about fluorescence lifetime imaging microscopy. When it is fluorescence lifetime imaging, that instrument can be used for doing fluorescence correlation spectroscopy. That's a very important and interesting technique. So with the help of that, you will be able to find out this property, like diffusion coefficient, etc., of some material or maybe some fluorescent sample. In that case, in fluorescence spectroscopy, the sample may not need to be fluorescent. Maybe you can study protein aggregation. Even this protein, like if it is not like fluorescent, Okay, so you can make use of this fluorescence correlation spectroscopy to look into this protein aggregation process or maybe biomolecular aggregation process. So these are all emerging things, and these are very useful and informative as far as scientific contents are concerned. Now we will just as promised in the last lecture, so we will just give you this this live cell image of 2-color live cell imaging of MDH in HEP G2 cells, and this HEP G2 cells transfected with this EGFP tubulin, these are shown in green, and were stained with MDH shown in cyan and imaged after every 30 seconds. So let's and it is taken from this reference. So let's try to see what is happening here. You see that nicely. It is, let me do it again. So you see that live cell imaging can nicely be done with the help of this time. I mean, it is not, I should not say time result. You can, doing the time slicing of the process. So you can see, so what is exactly happening inside the cell, that you can image with the help of this imaging technique using fluorescence microscopy. Okay, so now I will just get you the information about books that may be useful for your study, and occasionally I have consulted, in many occasions I consulted these books. Like the most important book is Principles of Fluorescence Spectroscopy by Lakovich. So it is from Springer. This is Wiley Visage, another very important book by Bernard Valeur and Santos. This Barbiran Santos. So it is molecular fluorescence principles and applications. This is very good book. This is also very useful, and it's a huge book. So not only just fluorescence spectroscopy, but fluorescence microscopy part, you can also get information about that in this book. Then another very good book is Principles and Applications of Fluorescence Spectroscopy by Albani. This publication you can go through. That is a very useful book. Then another is as far as photochemistry and photophysics are concerned, you can consult this book, and modern version of this book is also available with Turo, Ramamurthy and Siano. So that book you can consult. As far as basics of spectroscopy, you can consult this Modern Spectroscopy by Hollas, also Fundamental of Molecular Spectroscopy by Banuel and McCash. Regarding biomedical spectroscopy and imaging, you can go through this book, Light Spectroscopy, this book, Fluorescence Spectroscopy and Microscopy, this book, then Fluorescence Spectroscopy imaging tool, then Topics in Fluorescence Spectroscopy, edited by Lakovich, Laser Spectroscopy experimental technique by this author, then macromolecular sensing. So these are popular books, and you will be able to find many other books. Okay, if you go to a good library, then you will be finding a lot of books. And now I'll be giving photographs of some instruments, like taken from various sources, web sources. It is a Fluoromax 3. Although Fluoromax 4 version is available, Fluoromax 3, there is a fluorimeter, Horiba instrument fluorimeter. That's a very useful. I mean, it is just a view of a fluorimeter. Okay, this is your sample chamber. Okay, so this one, and also this is Parkin-Elmer luminescence spectrometer. Okay, this is the old version. Anyway, this is another instrument. Then this is Fluoromax 4. Then this is Fluorolog. It is a modular design. You see that many boxes are there. Modular design is sample chamber. So you can make it. Depending on your requirement, you can change these boxes. So this is light source, then it is optics, then you have sample, then you have detector. So Fluorolog is a very sensitive and good instrument as far as fluorimeter is concerned. Okay. Then you have this as far as lifetime instrument. This is the smallest one, is the TC-SPC by OBB. Originally it was from PTI. This is one lifetime spectrometer. Okay. This is another photograph of lifetime spectrometer, one from maybe Horiba, another from other company. Okay. You can find various companies who are selling, like Picoquant, then Horiba, then maybe PTI, and so on and so forth. Okay, maybe another is Edinburgh instrument. So these are the photographs of various imaging devices, like this is Olympus microscope. Okay, so this you see that you have your sample over here, and this is an inverted microscope. Then this is from Zeiss. This is an inverted microscope from Optica. Okay, and this is not an inverted, because you see that your objectives are over here. So this is not inverted, it is a regular. Okay, then this is a Nikon. That is from Nikon. Okay. Okay, and various websites may be useful. These are not the final references. You will be finding plenty of informations. Okay, one is like this one, another is this one, another is this one for ultrafast spectroscopy, and there are many other in many other sites where you will be finding plenty of information. And if you go through those websites and also many web resources, you will be able to enrich yourself about this particular thing. So the point is, whenever studying fluorescence spectroscopy, then you should consult the textbooks. I mean, may not be the list of textbooks I have shown over there. I mean, these are not the final. Okay, you can find other useful text in library, not only these foreign textbooks, but also these indigenous, I mean Indian textbooks sometimes are very useful. So you just keep watching on these books, and if you find that these are useful, you should go through it. And at the same time, these foreign textbooks and maybe web resources are very useful. You will be finding plenty of websites where where lots of information are posted there. And also maybe if you go through various literature, like various journals, then you will be finding plenty of information, especially this review journal like Chemical Review, then Advances in Physical Chemistry, Advances in Photochemistry, and so on. Like these review journals are very useful. Review journals are very useful in this regard. So you can go through these various review journals and pick up, depending on your necessity, whether it is just a fluorescence spectroscopy or whether it is a microscopy thing. Then you can go through those review articles based on your necessity. And if it is microscopy, then you can go through, there are competitive websites like from Zeiss, then maybe from Nikon, maybe from Leica, maybe from Olympus. Okay, so there are plenty of competitors in the microscopy thing. So if you go through those websites, you will be finding lots of information, especially like the parts of the instrument, how this instrument is run, what is the basic principle of that instrument, and then also this representative this imaging imaging files, that is the something is moving around, moving inside the cell, etc., etc. So you will be able to watch those if you go to that, if you click onto the particular website. So So while you know, studying this spectroscopy and microscopy, then you should go through those, and also not only those various company websites like various maker website. You are also encouraged to go through the websites of various renowned research workers, where you will be finding good number of informations and also their good publications. So you go through that and you read it, and I think you will be knowing a lot about the subject, and and also in knowing in order to know more about these instruments like this Fluoromax, etc., you are also encouraged to visit their websites, and a lot of informations are posted in their websites. Okay, in some websites they are also providing some representative data. So you are encouraged to go through those representative data, and then you study and see what is exactly happening. Like if you go to this OBB website or maybe similar websites, you will be finding this application notes, and also some of the representative publication relating to some of your questions. Okay, so you visit the websites depending on your question and depending on your requirement, and you should be able to find your answer. So while studying this fluorescence spectroscopy, you should go through you should visit various websites, you should consult various books, and these website list is not the final, it is just giving you idea of some of the representative research groups who are doing various type of research or involving fluorescence spectroscopy, like biogenesis function of mitochondrial membrane protein. So here basically you will be knowing application of fluorescence spectroscopy in using this mitochondrial membrane protein. Here single molecule fluorescence spectroscopy and microscopy of soft matter. Okay, so single molecule fluorescence means you need to have this microscopy business, okay, microscopy thing, okay, and also microscopy of soft matter. Now if you want to know like ultrafast spectroscopy, you want to know, then you click on the specific website or maybe specific group's website, where you will be finding plenty of information. Okay, so these are the way that you should organize yourself, that is organize yourself in studying, and I would urge you to look see all these different lectures. Total 40 lectures of under this fluorescence spectroscopy course, and if you have questions, if you have concern about this, then you should write me. So write me or you should contact this NPTEL website. You should write through NPTEL website. So you should be able to, as soon as I see them, then I should be able to or I will try to answer to your queries. So I hope that you enjoyed this series of lectures, series of 40 lectures, and I'll feel good that if while going through this series of lectures, if your interest about on fluorescence spectroscopy is increased, and also if you are if you learn something through this series of lectures, it will be rewarding to me. So I thank you all for your kind attention, and have good time. Thank you.